Hey everyone, welcome to the Basics lecture on neurons and membrane potentials. We're going to start this unit with the anatomy of a nerve cell called a neuron. This image shows one neuron signaling a second neuron. Please use chapter 48 in your book to label this diagram. Now let's take a close-up look at the membrane of a neuron. On the left is a sodium-potassium pump, and on the right is an open potassium channel. The sodium-potassium pump uses energy to pump sodium out and potassium in, just like we've learned previously, and the potassium channels open due to diffusion and let potassium back out again. Researchers can place small electrodes inside the cell and measure the difference in charge, positive or negative, across a cell membrane. The difference in charge is called the membrane potential. Because of the pump, sodium-potassium pump, the concentration of sodium is high outside, and the concentration of potassium is high inside. But because there's also the leaky potassium channels, the concentration of potassium is somewhat high outside as well. This means that the inside of the cell has fewer positive ions and is therefore more negative than the outside of the cell because we're comparing the two. And an electrode that measures the difference would measure about negative 70 millivolts. But the inside is more negative than the outside. This is called the resting membrane potential. So let's stop and see where we are. The pump and channel result in a plasma membrane that's slightly negative on the inside. You can see the little negative marks here, with lots of potassium inside, and the outside has lots of sodium and some potassium and is so slightly positive. Nerve cells use this difference in ion concentration in order to send signals from one end of the cell to another. The signal is called an action potential. If the membrane potential is made more positive on the inside, going from resting to more positive, this is called a depolarization signal. If the membrane signal becomes more negative, that's called hyperpolarization. And if the signal is already high and gets negative, that's called a repolarization signal. A strong depolarizing stimulus that causes the inside of the membrane to depolarize can sometimes make it depolarize up to a level called threshold. And at this threshold level, a cascade of changes causes a spike of depolarization and then repolarization that lasts just a few milliseconds, and this increase and then decrease is the action potential. In order for a neuron to start and send a signal down the length of the cell, we need to talk about 3 additional gated channels. Located mostly on the dendrites are ligand-gated ion channels, and there's many different types of these that have different effects on neurons. For our course, let's just say that a chemical from another neuron can come in and bind to the transporter and cause the channel to change from closed to open, and this would allow ions to rush across. In many cases, the one we'll consider, this will allow sodium to go from the outside of the cell to the inside. The other 2 channels are these: a voltage-gated sodium channel, which is closed at resting potential, but opens when the membrane depolarizes at about negative 60 millivolts, and then closes when the membrane reaches positive 40 millivolts. And there's a voltage-gated potassium channel, which is closed at resting potential, but opens at roughly 40 millivolts, and then closes when the membrane depolarizes down to negative 75 millivolts. Now let's follow the phases of an action potential. When the membrane is at its resting potential, all of these channels are closed, and the membrane is at its normal minus 70 millivolts. If a neurotransmitter comes and binds to the ligand-gated channel, causes it to open, and in this case, let's say sodium rushes in, then this causes the membrane to become less negative inside, to about minus 50 millivolts. This increase in the number of positively charged ions near these voltage-gated channels, remember that proteins have polar R groups, and the increase in positive ions nearby causes the R groups to respond, and that changes the shape of the protein. This opens the voltage-gated sodium channel on the nearby voltage-gated channels near the ligand-gated channel and causes more sodium to enter the cell. This makes the inside of the membrane less negative. If the potential increases enough to reach threshold, shown here, we go to the next step. A strong trigger opens all the sodium channels in an area. This causes a large rush of sodium into the cell, and the membrane becomes very positive inside compared to the outside, and you get this large depolarization spike. The positive potential inside the membrane causes the gated potassium channels to open and the sodium channels to close. You can see open potassium and closed sodium. This sudden change in permeability makes potassium rush out, makes the inside more negative, and the voltage returns towards resting, repolarizing. The potassium channels generally stay open a little longer than they need to, making the membrane slightly more negative than resting. This is called undershoot or hyperpolarization. This then corrects as all the channels close, and the sodium-potassium pump can return all the ions to their starting locations. This brings us back to resting state. All channels are closed, and the resting membrane potential is again at -70. Be sure you can describe all 5 of the stages in an action potential before you continue. Seen all at once, an action potential is essentially a sudden permeability of the cell membrane to ions, which rush in and then out for a few milliseconds, which is interesting but isn't useful in and of itself because we still need to get this change that might occur here to travel all the way down the length of the entire neuron. Luckily, the ions that rush in one part of the membrane can trigger the gates in nearby sections of the membrane to also respond. In this diagram, it shows that sodium that rushes in at one end of the membrane then has effects on either side and opens the next series of gates so that the depolarization moves farther down the membrane and farther still down the membrane. After our depolarization comes repolarization, shown here in green. Potassium ions rush out, and this wave travels all the way down the length of the axon, and behind it follows resting potential, returning the cell to normal. That's it for our video for today. Write out the answers to these questions to study for the basics quiz. Thanks for watching.